two forms particularly in the process of the utilization of the organizing, improve the health cares and also the fighting the disease, a branch of the biotechnology of the utilized in the field of medicines. It's also the include the productions of the medicines and the pharmaceutical product for the treating and also the diagnosis. There are so many examples, little bit, and also the examples of the things regarding the productions of the human insulin, the non-insulin sources, from the non-insulin sources, the productions of the hormones, even also the difference of the cytokines and the steroids, human growth hormones. These are also productions from the applications of the biotechnology. Gene therapy, also the like the hemophilia, even also the cystic fibrosis. The development of the vaccines, very interesting vaccines, we have already gained from these applications. And even also the drug production, pharmacogenomics, even also the gene therapy, tissue engineering, and the drug production. There are so many drugs produced by the applications of the biotechnological applications. And drug productions through the medicines, the isolations of the enzymes from the different plants, even also the animal, from isolated from the enzymes, the genetically engineering enzymes, and also the interferons. There are so many things, and even also the human growth hormones, and also the pharmacogenomics, that is the pharma, that's in the drug or the medicines, and also the genomics, the study of genes, already you know. Then the future future of the pharmacogenomics, this is very much important. And by 2025 include the most information related to the drug metabolizing enzymes are to be produced by applications of the biotechnology. And after that, by 2050, then, and also it mitigate the include the an entire individual genomes. These are the today and tomorrow. Then today we have response to the drugs 30 to 60%, and also the does not response to the drugs everyone else. আমরা দেখি মাত্র কেবলমাত্র 8%, but the tomorrow by the applications of the biotechnology, the 100% will be success rate. Gene therapy, even also the gene therapy of the delivery systems, and these are the direct delivery system and cell mediated delivery system of the applications of the biotechnology. And this is the therapeutic gene after and directly the therapeutic gene is packed into a delivered vehicle such as a retrovirus directly, and even also the cell mediated cell based delivery system like the embryonic stem cell delivery system, even also the others. These are the method tissue engineering. There are so many things in the tissue engineering. The genetic testings, the different disease and the disorder, but detect by the applications of the DNA molecule to determine his and her sequence from the mutant genes, like when people suffering in different types of the new disease like corona, like different types of the influenza, always changeable day by day, year by year, month and month. Then the detect by the sequencing technique. The genome also the individuals of the scanned in the purpose of the scientists regarding the mutation detections by different way. These are the very important thing, PGD, even also the PND, and also the pre-symptomatic testing, even also the confirmational diagnosis, and the forensic, even also the identified testing. These are the applications of the detections in the health. Cytogenetics, even also the molecular diagnosis. There are so many types, and particularly these are the ways. Then the already you know about the process of the typing, the karyotyping technique, even also the fluorescence techniques in the in situ, hybridizations, even also the molecular diagnostics, and also the detections of the new mutations. And also the next generation sequencing, even also the hetero duplex analysis. Now, very few work has been done in our laboratory, very small laboratory developed by Dr. Kaushik Das. And that time Kaushik Das is was my student as well as my colleague in the Gop College, Rajanaranda Lal Khan Women's College. First I have seen in a hospital established in 2002 in Midnapur town, mainly for treatment in kidney failure disease. I have faced so many things regarding the patients when come or came into the dialysis center, as because hemodialysis, peritoneal dialysis can't till not to cure the end-stage kidney diseases. That's why there are so many models developed in rat model. Partially dehydration. When people are dehydrated, then kidney is failure. When people suffering in the diabetes, kidney is failure. When people suffering from the cardiac disease, people suffering the kidney disease. So many. The end-stage kidney disease is very much important things. That's why the CKD 10% people are affected in the chronic kidney disease nowadays, but not to detect properly and right time. এখনো পর্যন্ত রাইট টাইমে কিডনি ডিজিজ ধরা পড়ে না। এখনো পর্যন্ত ডিটেকশনের প্রসেস যা রয়েছে, তখন কিডনি ডিজিজের ক্ষেত্রে আমাদের যে মার্কারগুলো আছে, বায়োমার্কার, দ্যাট উইল বি এনহ্যান্স। Even also sometimes people are suffering for end stage, not to cope up or the cure from the previous life, and they are waiting for die. Even also my dear students in the year 1987 in Midnapur College Physiology students. Name is Saurav from the Gwaltor. All the things in the modern way of the treatment of the kidney disease, like transplantation from mother, donor is the mother, his mother, even also the hemodialysis, peritoneal dialysis, not to cure. And after suffering long time, he died. These are the scenario. Not even anywhere in throughout the country. These are the very gloomy situations in our country, in the medical field. That's why we work in different way regarding the, even also when you consume more paracetamol, like acetaminophen, painkiller, like that, you also suffer sometimes in the kidney disease. There are so many factors. I do not discuss elaborately. And we we searching the alternative strategy. What are the alternative strategy? Then we developed in our small laboratory, small facilities, and phytocompounds from the plant product, that the herbal product. The Kaushik first work in the Terminal Urjuna, Uthania, Sominifera, like that. Even also the bacteriotherapy. These are the very success rate in the bacteriotherapy is very much important, as because our gut microbiome not to produce the urease enzyme in human beings. That's why only the product produced by the gut microbes, those are the probiotics. And we have isolated the new bacteria, then administered to the kidney suffering rat, and the promotions of or the cure the rat particular in the different way. And before that, so many things we have established in the experiments. These are the very much important. These are the conventional method, and regarding the kidney management, diabetes, hypertension, like that. The alternative strategy of the bacteriotherapy, we have isolated the new bacteria ADK10 and ADK14. It are 2010. We have isolated from the cheek, stomach, and established as a probiotics. And that probiotics that produce so many urease enzyme. That enzyme are fate of urea. And also the ADK10. ADK, our research scholars, particularly now he is a assistant professor in which college? Asutosh College in Kolkata. Dr. Orpita Mondal. He was also the inspired fellow like Saboni and Suchismita. Then ADK, D means DN or Dilip Nandi or like that Dilip Kumar Nandi, whatever may be. K means Keshav. Another my very pride, and I am very much proud, my first stage of students. Then the name is, these are the name, and isolated characterizes and isolations and characterize, these are the bacteria. And even also we have go for different techniques, applying different techniques regarding the same, the adhesive powers of the bacteria, like that we have that probiotic, even also the cell line, different cell line, also we have studies of the adhesion property of that particular probiotics, and establish as a not in biochemical only, then establish as a probiotic bacteria ADK10, even also the so and so things. Then another in the nutraceutical nowadays in the form of capsules or the different types of the bar. That bar supplied from the DFRL, Defense Research Institute of Food, Mysore, and particularly from collected the quercetin, isolated extractions from the plant product materials, then prepared this type of the bar, composite bar or the laddu, regarding the form of the laddu, and administers to the stressful conditions of the army, even also in our college students during the period of corona. We have a massive results and very important results after the administrations of that. I do not elaborate discuss about, sir, our time ache amar? Nahi, acha thik ache, chhod dicchi. Next, these are the methods, you know, so many things. HPLC done by Suchismita, and with the help of Suchismita, and the facts and etc. The same structures from the other things. I do not discuss anymore. Any questions from the students? No, apnar question আমি নিতে পারবো না. Not a single question. Please, I do not accept your question, sir. I do not accept, as because you are a veteran teachers, you are very much knowledgeable. I do not accept your. Please, student, I do not accept, sir. No, ami doctor no, sir. You go to the doctor. Please, students, no question. No question. আরে কিছু তো বলো না, আমি নিতে পারবো না, না আমি অ্যান্সার দিতে পারবো না, আমি স্যার তুমি পারবে, তুমি চলে এসো, ইউ আর দ্য ইন্টারন্যাশনাল ফিগার, ইউ মে কাম। থ্যাংক ইউ এভরিওয়ান। মে আই রিকোয়েস্ট ডক্টর কৌশিক দাস, প্লিজ কাম and finished within a few minutes. Kaushik, please.
দুটো আছে আবার, হ্যাঁ এইটাই। we are going to felicitate Dr. Kaushik Das, Assistant Professor, Department of Nutrition, Belda College. Our madams will felicitate, sir.
I just introduced Dr. Kaushik. Dr. Kaushik actually passed out from the physiology in Vidyasagar University and Midnapur College. After that, done his PhD in Vidyasagar University under my supervision, even also the Professor Devidas Ghosh both. And Kaushik really done very nicely in 2010, and that triggers with converted myself in the research in 2010, not more than before that. And Kaushik after that as a nutrition teachers in our college, as a coordinators in charge, after that and a permanent teachers substantial post in assistant professors in Belda College. He has a lot of things regarding any types of the project like that, even also the others, so that students are enjoy his lectures. I think you may finish. Time, sir? 10 minutes.
হ্যাঁ 10 মিনিটে হয়ে যাবে। হ্যালো হ্যালো গুরুদেবও নম। প্রথমে আমি আমার গুরুদেব ডক্টর দিলীপ কুমার নন্দী স্যারকে আমার অজস্র প্রণাম জানাই, কারণ আমি আজকে এইখানে যে লেকচারটা দিতে এসেছি তার জন্য কিন্তু স্যারেরই সম্পূর্ণ অবদান আমার প্রতি। এবং সবাই আমরা জানি যে প্রত্যেক পুজোতেও আমরা শুরু করলে আমরা কিন্তু গুরু ব্রহ্মা গুরু বিষ্ণু গুরুদেব মহেশ্বর ওইটা করেই আমরা পুজো শুরু করি। অতএব গুরু সবার মাথার উপরে। এবং তারপর আমি এই কলেজের ডাইরেক্টর ডক্টর প্রদীপ ঘোষ স্যার, প্রিন্সিপাল ডক্টর সুদীপ্ত চক্রবর্তী স্যার, আমার ফ্রেন্ড ডক্টর শান্তনু দাস মহাপাত্র, এবং আমার স্টুডেন্ট কাম কলিগ শ্রাবণী, সুচিস্মিতা, মনীষা এবং আরও সবাই, এবং প্রিয় ছাত্রছাত্রী, আজকে যে প্রেজেন্টেশনটা আমি আজকে অ্যাকচুয়ালি করছি এর উপরে আমার লাস্ট প্রায় আগে তো স্যার বললেন যে আগে আমরা কিডনি ডিজিজে, আমার পিএইচডি কাজ হচ্ছে কিডনি ডিজিজের উপরে। এরপরে আমি দেখলাম যে কিডনি ডিজিজের উপরে কাজটা খুব কঠিন আর কি, মানে আমাদের যে এই ল্যাবরেটরি সেট আপে কিডনি ডিজিজের কাজটা এখনও সুচিস্মিতা ম্যাডাম অবশ্য চালিয়ে যাচ্ছে, কিন্তু খুবই কঠিন এই কারণে যে কিডনি ডিজিজের পেশেন্টরা কিন্তু একটা জায়গায় এসে এন্ড স্টেজ রেনাল ডিজিজে এসে কিন্তু ওরা আর সেটা কিডনিটা রিভার্স হয় না, ইরিভার্সিবল থেকে যায়। সেই জন্য আমি দেখলাম যে এই কিডনি ডিজিজের উপরে যদি আমি রিসার্চ করি তাহলে এটার বেশি এগানো যাবে না। সেই জন্য রিসার্চটাকে আমি ডাইভার্ট করলাম বা ট্রান্সপোর্ট করলাম যেটাকে ডায়াবেটিসের উপর। ডায়াবেটিসের উপরে রিসার্চ করতে গিয়ে দেখলাম যে কি না এখন তো প্রচুর পরিমাণে সব ফাইটো কম্পাউন্ড মানে রাস্তায় যেতে যেতে হুমড়ি খেয়ে পড়ে যাচ্ছে, এই একটা নতুন গাছ, নতুন প্ল্যান্ট। তো সেই হিসাবে দেখতে দেখতে আমিও দেখলাম যে মানে অজস্র, মানে আইসিএমআর তো প্রায় কয়েক হাজার প্ল্যান্টকে পুরো এস্টাবলিশড করে রেখেছে অ্যান্টি, মানে অ্যাজ এ অ্যান্টি ডায়াবেটিক অর অ্যান্টিবায়োটিক ঠিক নয়, মানে বায়োঅ্যাক্টিভ অর ফাইটো কম্পাউন্ড। তারপর বিভিন্ন সায়েন্টিফিক কমিউনিটি, বিভিন্ন ধরনের ফ্রেন্ড, স্যার, স্টুডেন্ট সবার সঙ্গে আলোচনা করতে করতে একটা জায়গায় আমরা দেখলাম যে ডায়াবেটিসটা এখন একটা, আমরা সবাই জানি ডায়াবেটিস সম্বন্ধে, তো এখন কি বলবো, শহরে প্রত্যেকটা ঘরে দেখলেই একজন করে ডায়াবেটিস, এবং ডায়াবেটিস মেলাইটাস, যেটা ব্লাড গ্লুকোজ হাই, যেটা হচ্ছে আমাদের এই এখানেও যে হেড অফ দ্য ইনস্টিটিউট ডক্টর প্রদীপ ঘোষ, ওনারও ডায়াবেটিস। ডায়াবেটিস 110 above 110 milligram per dl, that is pre-diabetic, and 126 fasting, that is diabetes. একবার হয়েছে মানে আর কিছু চিন্তা নেই। তো নেক্সট সেই হিসাবে ডায়াবেটিসের উপরে কাজ শুরু করার পরে, যেটা আমি প্রায় 4-5টা পেপার পাবলিকেশনের পরে বা বিভিন্ন জায়গায় প্রেজেন্টেশনের যখন আমি যেতাম, সেখানে একটা কোশ্চেন কিন্তু আমি খুবই ফেস করেছি, যেটা হচ্ছে যে তুমি যে মডেলটা করেছো সেই মডেলটা কি, টাইপ 1 ডায়াবেটিস না টাইপ 2 ডায়াবেটিস। আমরা জানি যে আমাদের এমনি জেনারেলি ডায়াবেটিস হচ্ছে 4 প্রকার: টাইপ 1, টাইপ 2, টাইপ 3, টাইপ 4। মেজর টাইপ 1 যে ডায়াবেটিস, সেটা এটা আমি আর বেশি লিটারেচারে যাচ্ছি না। টাইপ 1, টাইপ 2, টাইপ 3 হচ্ছে এমনি হরমোনাল ডিসঅর্ডারে হয়, আর টাইপ 4কে বলা হয় জেস্টেশনাল ডায়াবেটিস। এখন আইসিএমআর যেটা বলছে যে 4 প্রকার ডায়াবেটিস। তো টাইপ 1 ডায়াবেটিস আর টাইপ 2 ডায়াবেটিস হচ্ছে, টাইপ 2 ডায়াবেটিস হচ্ছে মানে টোটাল কেসের 90 থেকে 95% হচ্ছে টাইপ 2 ডায়াবেটিস। তো আমরা যে অ্যানিম্যাল মডেলে যে কাজটা করি, সেটা টাইপ 2 না টাইপ 1, এটাই হচ্ছে বিগ কোশ্চেন। সেই কোশ্চেন, ওই রিসার্চ, কোশ্চেন ছাড়া তো রিসার্চ হয় না, এই কোশ্চেনের উপর অ্যান্সার খুঁজতে খুঁজতে আমি একটা ছোট্ট কাজ করলাম, সেটাই আজকে আমি প্রেজেন্টেশন করছি। যে হাই লিপিড ডায়েট অ্যান্ড স্টেপ্টোজেরক্সিন ইনস্টিগেটেড টাইপ 2 ডায়াবেটিস mellitus in rats, challenges and issues. We all know the type 2 diabetes T2DM is the short form, which account for over 90 to 95% of all diabetes. At present, there are no curative treatment available type 2 diabetes. So the urgent need for prospering new preventive and therapeutic strategy to combat T2DM. By reasons of the progression of this disease, clinical animal model required to assess the pathogenesis of disease, which will reflect an equivalent to human. And so objectives of this model, ceftazidime is a drug, can be used for induction of both type 1 type 2 diabetes. We all know that, and literature said that. An animal model which incorporate a high lipid diet to induce peripheral insulin resistance, followed by low dose ceftazidime to target the type 2 diabetes mellitus rat. So we know that when we consume the diet, every sort of diet, আমরা যখনই যা খাচ্ছি, ব্রেকফাস্ট খাচ্ছি, একটা বিস্কিট খাচ্ছি, লাঞ্চ খাচ্ছি, ডিনার খাচ্ছি, যে টোটাল গ্লুকোজকে যদি আমরা 100% ধরি, টোটাল গ্লুকোজ 40% অফ গ্লুকোজ stored in the as a glycogen in the liver first, যখনই আমরা যাহা খাচ্ছি। Then 30% glucose utilized for BMR, basal metabolic rate, and next rest of the 30% if physically active, যদি আমরা ফিজিক্যালি অ্যাক্টিভ থাকি, ওয়াকিং করি, লাফালাফি করি, গান করি, হামাগুড়ি দিই, তখন কি হবে না, সেই 30% যে গ্লুকোজ মায়োসাইট, দ্যাট মেনলি স্কেলিটাল মাসল or few cardiac muscle utilized that glucose. If we are inactive, physically inactive, 30% attached to adipocyte, that means fat cell. যদি আমরা inactive থাকি, তাহলে কি হচ্ছে, the insulin receptor present in our throughout the body, all over the body, but insulin receptor main that function the glucose transport mainly occur in the skeletal muscle, that is the myocyte, and fat cell, adipocyte, and little amount of cardiac cell that present the GLUT4, glucose transporter 4, and any other our body, brain cell, another cell, kidney cell, GLUT2, GLUT1, GLUT3, GLUT4, GLUT4 except GLUT4. Only GLUT4 present in the skeletal muscle cell and GLUT4 present in the fat cell. If skeletal muscle cell uses the glucose, okay. If skeletal muscle cell does not use the glucose stored as a fat in the adipose tissue. Then type A diabetes, type 1A, actually type 1 diabetes nowadays two types. Actually mean white person and black person, that is another part. So type 1 diabetes, we actually called the genetically modified diabetes, that is the type 1A diabetes mellitus, T1ADM. Actually there are a triangle cause when anti-islet and anti-islet autoantibodies are produced. Then when a microorganism, if that is the genetically modified, we all know that HLA, human leukocyte antigen class 2 molecule immune dysfunction, HLA gene dysfunction, that is carried out the inherited manner. Then বলছে যে যদি আমাদের যে HLA জিন মানে ম্যাক্রোফাজ মনোসাইটের ভিতরে যে জিনটা, সেই জিনটা হচ্ছে যদি মডিফাইড হয়ে থাকে, মিউটেন্ট হয়ে থাকে, তখন হলে কিন্তু আমাদের কোনো সমস্যা নেই, মানুষ সুস্থভাবে বেঁচে থাকতে পারে, টাইপ 1 ডায়াবেটিস হবে না, যতক্ষণ না ওই পার্টিকুলার ভাইরাস, ব্যাকটেরিয়া বা প্রোটোজোয়া আমাদের বডিতে ইনভেট করছে। বডিতে যখনই মাইক্রো অর্গানিজম ইনভেট করবে তখনই কি হবে না, এই HLA ওই ম্যাক্রোফাজ, কারণ আমরা জানি যে ম্যাক্রোফাজ ও মনোসাইট হচ্ছে ভার্সেটাইল অফ নেচার, যখনই যে অ্যান্টিজেনটা ভাইরাস ব্যাকটেরিয়া আসে তাকে তার মতো করে প্রসেসিং করে। পার্টিকুলার দেখা গেল কিছু হয়তো একটা সাসপেক্ট আছে কোনো একটা মানুষের টিবি, টিউবারকুলোসিস ব্যাকটেরিয়া যতক্ষণ না হবে তার ওই টাইপ 1 ডায়াবেটিসটা হবে না, মানে ওই HLA জিন অ্যাক্টিভেট হবে না, তার ফলে কি অ্যান্টি আইলেট অটো অ্যান্টিবডি উৎপন্ন হবে না। তো অ্যান্টি আইলেট অটো অ্যান্টিবডি কী করে না, বিটা সেল, এটা হচ্ছে কি নর্মাল আইলেট, these are the beta cell, pink color, and another is the alpha cell, mainly alpha and beta cell. When anti-islet autoantibody attack, the beta cell are destroyed, and here the beta cell are not present. That is the type 1 diabetes. Next, type 2 diabetes is simple form. Type 2 diabetes, insulin bind the myocyte, skeletal muscle cell, insulin receptor. If insulin attracts more to binds with its receptor present in the adipocyte than myocyte. Actually insulin প্রচুর আসক্তি হচ্ছে কি যে ফ্যাট সেলে বাইন্ড হওয়ার, যদি অতিরিক্ত পরিমাণে ফ্যাট সেল বাড়তে থাকে তাহলে কি হয়, স্কেলিটাল মাসল সেলে ইনসুলিন আর বাইন্ড হয় না, তার ফলে কি হয়, স্কেলিটাল মাসল সেলের ইনসুলিন ডিসেন্সিটাইজড টু বিকাম ডিসেন্সিটাইজড টু ফার্দার বাইন্ড টু ইটস রিসেপ্টর insulin. So if the myocytes are not accept the insulin, then adipocytes are huge amount of adipocytes are fat cells are increased and insulin receptor increased. So if the insulin receptors are increased in the fat cell, এই যে ভুঁড়ি হয়ে যাচ্ছে, মোটা হয়ে যাচ্ছে, ঠিক আছে, তাহলে কি প্রচুর পরিমাণে ইনসুলিন রিসেপ্টর এখানেই হয়ে যাচ্ছে, স্কেলিটাল মাসলে আর কতটুকু আছে, তো স্ট্যাগন্যান্ট, তাহলে স্কেলিটাল মাসল সেলে কি হচ্ছে, ইনসুলিন আর বাইন্ড হচ্ছে না, সেই জন্য কি হচ্ছে, ফ্যাটিক হচ্ছে। Then this is the healthy individual, beta cells are present. Beta cells, actually beta cell called beautiful lady, মানে একটা সুন্দরী মহিলা, তার চারদিকে আলফা সেল, গ্লুকাগন প্রডিউসিং সেল, এই যে এগুলো বিটা সেল, পাশে হচ্ছে কি আলফা সেল। তো বিটা সেলস আর partially damaged in the type 2 diabetes, partially damaged. Then insulin secretion and action, actually here insulin bind when the blood glucose level increase, when we consume any diet, blood glucose level increase, GLUT2, glucose transporter 2, that is the two-way transporter, when beta cell increase the glucose outside from beta cell to blood, ECF, when ECF glucose high enter, glucose enter in the cell, glycolysis, ATP, and then insulin secretion. This is the insulin secretion and that when insulin bind to the insulin receptor substrate that phosphorylated, and via the AKT protein activation, the GLUT4, actually here a new idea, new science that called the GLUT4 are not produced in our cell. GLUT4, when in the intrauterine life, GLUT4 are already present in the cytoplasm. When insulin bind to its receptor, just AKT translocate the GLUT4 from cytoplasm to membrane to transport the glucose for activation of the skeletal muscle cell for our activities. Then insulin action with receptors, and there are many proteins I not discussed there. Specific, this is my research, specific T2DM sensor. তাহলে আমরা এখন কি বলবো যে কোন আমরা টাইপ 2 ডায়াবেটিস স্টেপ্টোজোটক্সিন দিচ্ছি, ইনজেকশন দিচ্ছি অ্যানিম্যাল মডেলে, তো সেটা কি হচ্ছে না হাই ব্লাড গ্লুকোজ, তো দ্যাট if we called type 1 or type 2. Now type 2 কখন বলবো, we already discussed many diabetologists, and that is the specific type 2 diabetes mellitus sensor is the C peptide. C peptide, when we all know that proinsulin is synthesized, and then proinsulin to insulin C peptide, one molecule insulin and one molecule C peptide, and that C peptide are not utilized in our body. That C peptide just excreted through the after 24 hours through the urine. Then so C peptide is a substance that is created when the hormone insulin produced and released into the body. The C peptide test can help determine the type of diabetes a person has and how much insulin their pancreas is producing. And another specific T2DM sensor is the pancreatic glucokinase, because glucokinase, if glucokinase is activated and there is in the beta cell there is no hexokinase, only glucokinase, and that glucokinase is the main sensor of insulin secretion. This is the directly proportional. So glucokinase in the beta cell serves as a glucose sensor, amplifying insulin secretion as blood glucose rises. Glucose transported by GLUT2. And research questions. What are the research questions? The most common chemical used to induce diabetes is tetrodotoxin. It can be used for induction of both type 1 and type 2. High dose of STZ severely impair insulin secretion, a feature that is similar to type 1 diabetes. Means when type 1 diabetes progressing, there is no beta cell, there is no insulin in the blood. And on the other hand, low doses of streptococcin causes mild impairment in insulin secretion that is more closely resemble in the later stage of T2DM. মানে যখন যেমন আমার মায়ের এখন ইনসুলিন ছাড়া হয় না টাইপ 2 ডায়াবেটিস, মানে কি একটা মায়ের কত বছর হলো না, প্রায় 20 বছর পরে দেখা যাচ্ছে যে ইনসুলিন ছাড়া সে আর বাঁচবেই না, কোনো ওরাল হাইপোগ্লাইসেমিক এজেন্ট অর ডায়াবেটিক মেডিকেশন কোনো কাজই করবে না, কারণ সেখানে বিটা সেল কী হয়েছে, কম হয়ে গেছে, ইনসুলিন খুব কম পরিমাণে সিক্রেশন হচ্ছে। However, the low dose streptococcin model does not address the insulin resistance axis typically seen in the T2DM. So some previous studies have indicated that animals that were overfed a high lipid diet developed insulin resistance. Therefore, a model which incorporate a high lipid diet to induce peripheral insulin resistance followed by low dose of streptococcin and beta cell would closely mimic the not only phenotype but also pathogenesis of human T2DM. To find out the answer from this research question is to present feature of frequently used model T2DM in rat induced by HLD, high lipid diet and low dose streptococcin. So that is the experimental design. We actually this experimental design and this experiment was conducted in the Raja Anil Khan Women's College Animal Ethical supported. So there is two group, control at 10, there is a control group fed only normal diet, control group fed only HLD diet, and STZ injected rat fed HLD diet and STZ injected rat fed normal diet. মানে এই চারটা গ্রুপ, এখানে STZ 20 মিলিগ্রাম পার কেজি বডি ওয়েট, 40 মিলিগ্রাম, 60 মিলিগ্রাম, 80, বাট এখানে কি হয়েছে আমরা HLD ডায়েট দিয়েছি, মানে হাই লিপিড ডায়েট দিয়েছি, বাট এখানে হচ্ছে নর্মাল ডায়েট দিয়েছি। Here after STZ injection we can see that after 3 days or 72 hours the blood glucose, all STZ injected rat blood glucose high to the 600, near about 600, and then throughout the, it is not a treatment, not a treatment experiment, just only experimental. Here the blood glucose, there are various doses of the STZ and recovery of the blood glucose from itself rat. Next, the hemoglobin A1C level. Here the 40 milligram normal diet is the 60 milligram normal diet, HB hemoglobin high, but the 40 milligram HLD diet is this is the target our T2DM. And plasma insulin level, here insulin level 2 nanogram per ml, 0.5 nanogram per ml. The literature and William endocrinology books also another literature said that proposed that 0.5 below 0.5 milligram 0.5 nanogram per ml is actually exist in the type 1 diabetes, the plasma insulin level. And also plasma C-peptide level, it is also 0.1 nanogram per ml C-peptide level indicates the type 1 diabetes. So here 40 milligram and 60 milligram streptozotocin normal diet, the C-peptide level below the 0.1 nanogram per ml. But here when HLD diet rats, surprisingly the above the 0.2 nanogram per ml. That indicated. And also pancreatic glucokinase, total cholesterol, here high HLD diet, total cholesterol and total triglyceride also high. So it authenticate to HLD and hyperlipidemia, and it is the histological study, and it has been hypothesized that the combination of HLD and 40 milligram STZ per kg body weight treated rats serves an authenticate animal model for T2DM without any contradiction.
Thank you.
Thank you.
Thank you, Kaushik, for your
যদি কোশ্চেন হয় ভালো হয়, কারণ আমারও মানে এটা
excellent. যদি কেউ কোশ্চেন করেন করতে পারেন। আর কেউ আছে? হ্যাঁ বলো 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 বলো, কুইক। মাইক্রোফোন নেই? তোমার কোন কলেজ বলবে? চলে গেছে।
Good morning, sir. I'm Medinapur City College Food Science and Nutrition student. So my question is, why you choose high lipid diet and streptozotocin in type 2 diabetes mellitus?
Actually it is my work, high lipid diet and streptozotocin, because when streptozotocin injected in the peritoneally to the rat, streptozotocin is a toxic for the beta cell, particularly beta cell, because streptozotocin ট্রান্সপোর্ট, কারণ প্রত্যেকটা তো আমাদের ড্রাগ হোক যাই হোক তাকে তো ট্রান্সপোর্ট হতে হবে সেলের ভিতরে। GLUT2 মানে গ্লুকোজ ট্রান্সপোর্টার 2 যে প্রোটিন, সেটা হচ্ছে বিটা সেলে থাকে, কিডনি সেলেও থাকে, লিভার সেলেও থাকে, কিন্তু স্ট্রেপ্টোজোটক্সিন বিশেষ করে GLUT2টা বেশি থাকে হচ্ছে বিটা সেলে। স্ট্রেপ্টোজোটক্সিন যখনই আমরা ইঞ্জেকশন দেবো, GLUT2 দ্বারা স্ট্রেপ্টোজোটক্সিন হচ্ছে বিটা সেলের ভিতরে প্রচুর পরিমাণে ঢুকে যাবে, এবং স্ট্রেপ্টোজোটক্সিন just destroy the DNA in the beta cell and necrosed the beta cells to cause the decrease the beta cell, and decreasing the beta cell, that is the decreasing the insulin level. Actually high dose of streptozotocin causes total necrosis of the beta cell, মানে 100% beta cell necrosis করে দিচ্ছে। সেই জন্য কি হচ্ছে যে আমরা যখন HLD ডায়েট যখন দিচ্ছি, হাইপারলিপিডেমিক ডায়েট বা হাই ফ্যাট ডায়েট যখন আমরা খাওয়াচ্ছি, তখন কি হচ্ছে ফ্যাট সেল প্রচুর পরিমাণে বাড়ছে। আর আমরা কি দিচ্ছি এখানে, আমরা 40 মিলিগ্রাম বললাম, আমরা তো এখানে একটা দেখাইনি, আমরা 80 মিলিগ্রাম পার কেজি বডি ওয়েট স্ট্রেপ্টোজোটক্সিন দিয়েছিলাম, 100 মিলিগ্রামও দিয়েছিলাম। জাস্ট 3 দিনের মাথায় সব র্যাট মানে 80 মিলিগ্রাম পার কেজি বডি ওয়েট আর 100 মিলিগ্রাম সব মরে গেছিলো র্যাট। 60 মিলিগ্রামে বেঁচে ছিল, 40 মিলিগ্রামে, 20 মিলিগ্রামে যেমন কিছুই হয়নি নর্মালের মতো। দেখা গেল HLD হাই লিপিড ডায়েট যে র্যাটগুলোকে দিয়েছি 60 মিলিগ্রামে কিন্তু তাদের প্রচুর পরিমাণে বিটা সেল লস হয়েছে। কিন্তু শুধু দেখতে পাচ্ছি যেটা 40 মিলিগ্রাম মানে এটা হচ্ছে লো ডোজ। লো ডোজ মানে হচ্ছে জাস্ট লাইট ইম্পেয়ারমেন্ট, পার্সিয়ালি ইম্পেয়ার্ড, পার্সিয়ালি নেক্রোস দ্য বিটা সেল। মানে হ্যাঁ সেইটাই তো স্যার কাজ। টাইপ 1 বলছি না, কারণ টাইপ 2 বলছি, কারণ আমার বিটা সেল এখানে আছে, ইনসুলিন আছে। যদি আমার ব্লাডে ইনসুলিন থাকে নর্মালি, সে তো সব বিটা সেল মানে টাইপ 1-এর ক্যারেক্টারিস্টিক হচ্ছে স্যার ইনসুলিন জিরো, ইনসুলিন জিরো, বেসাল লেভেল অফ ইনসুলিন মানে ফাস্টিং বেসাল লেভেল ইনসুলিন পুরো জিরো থাকবে, মানে হ্যাঁ, কিন্তু
পার্সিয়ালি
না পুরো জিরো হচ্ছে না, কিন্তু যখনই
কমছে না।
এইচএলডি ডায়েট দিচ্ছি, লো ডোজ এসটিজেড দিচ্ছি, তখনই কমছে। না, নাম্বারও বেড়ে যাবে স্যার অ্যাডিপোসাইটস। হ্যাঁ হ্যাঁ।
ঠিক আছে, থ্যাংক ইউ। Paucity of time, no questions to for the session. Any questions you outside the seminar? Due to the paucity of time. Okay, over to the coordinator. Go.
Thank you to our respected presenters. Now we are moving on to the valedictory session and the award giving ceremony. First, we will move to UK Ghosh Memorial Young Scientist Award. And for giving the award, we are requesting Dr. Pradeep Ghosh, Honorable Director of Mednapur City College, to come to the stage.
I am going to announce here the names of the recipients of the Young Scientist Awards. The first name I am going to take here of the recipient of Young Scientist Award is Dr. Kuntal Ghosh. Sir, please come to the stage.
A big round of applause, everybody.
The second recipient of Young Scientist Award is MD Rehan Uddin. Sir, please come to the stage. On behalf of him, if anybody is interested, please come to the stage. Okay. Any student from Raja Enel Khan Women's College, please.
Thank you, sir. Next is the award for oral presentation of research scholars. And to give the award, I am requesting Dr. Sudipta Chakraborty, Principal, Mednapur City College, to come to the stage.
We are going to announce here the names of the winners of the oral presentation of research scholar. The first position holder is Ria Kaur, research scholar, Mednapur City College. Ria, please come to the stage. Here we have two names as second position holders. The first name is Pipika Das, research scholar, Mednapur City College. And the next name is Purna Nondi, research scholar, Raja Enel Khan Women's College. Pipika and Purna, please come to the stage.
Thank you, sir. Next, we are moving to PG Poster Presentation Award. And to give the first prize, I am requesting Professor Nirmal Kumar Sarkar to come to the stage.
Now it's time to take the name of the rank holders of PG poster presentation. The first name is Sunit Kumar Khas and his team, Mednapur City College. You please come to the stage.
Thank you, sir. Now we are moving. Now we are moving to the second prize, and to give the second prize, I'm requesting Professor Dilip Kumar Nondi to come to the stage.
The name of the second position holder is Jhili Karan and her team. Department of FSN, Midnapur City College. Please come to the stage.
Thank you, sir. Now we are moving to the third prize. And to give the third prize, I'm requesting Dr. Kuntal Ghosh, Vice Principal, Midnapur City College, to come to the stage.
I am going to take here the name of the third position holder of PG poster presentation, Suraj Das and his team. You please come to the stage. MVOC Technology, Mukbedia Gangadhar Academy. Okay, sorry, Academy. You please come to the stage. Mahavidyalaya. Okay, on behalf of them, Pranati Bera is taking the award. You please come to the stage.
Thank you, sir. Now we are moving to the UG Poster Presentation Award, and to give the first prize, I am requesting Dr. Shantanu Kaur Mohapatro to come to the stage.
Now it's time to take the name of the first position holder of UG poster presentation. The first name is Ria Kander and her team, Raja Enil Khan Women's College, BMLT 4th semester. You please come to the stage. Okay, on behalf of her, Pranati, Purna. Okay. Okay, Purna is on behalf of her Ria. Purna is taking the certificate.
Thank you, sir. And now for the second prize, I'm requesting Dr. Suchishmita Roy to please come to the stage.
I am going to take the name of second position holder, Puspita Shaw and her team, Medical Microbiology, Midnapur City College. Puspita, please come to the stage. On behalf of Puspita, Souvik is taking the certificate.
And now for the third prize, I'm requesting Dr. Shravani Pradhan to come to the stage.
The third position holder in this event is Snigdha Adhikari and her team, Department of Microbiology, Midnapur City College. Snigdha, you please come to the stage.
Thank you, everyone. Now this is a moment to reflect on the insights gained, express our gratitude to all the contributors, and celebrate the success of this enriching event.
Follow recipients of the Nutrition Month celebration.
Now we are calling the names of the recipients of Nutrition Month celebration, and to give the award, I'm requesting Dr. Kaushik Das to come to the stage.
The
Sir.
first, the first position holder in this event is Rajashri Maji and her team. Please come to the stage.
UG.
UG. এটা কি PG? UG এরপরে
এটা UG পুরোটাই
আচ্ছা আচ্ছা আচ্ছা
কৌশিক দাই দাদা এটা PG দেখে দাও সেকেন্ড
The name of the second sorry.
sir please stay in the stage.
The name of the second position holder in this event is Mousumi Samanto and her team. Please come to the stage.
First second ঠিক আছে
Nutritional celebration PG students.
Now we are moving to Nutrition Month celebration for PG students, and I'm requesting Dr. Kaushik Das sir to stay on stage and give the award.
The name of the first position holder in this event is Anona Samanto and her team. Anona, please come to the stage.
Please be quick, as we are due for our 9th Foundation Day celebration.
The second name is Pooja Pal and her team. You please get ready. Pooja Pal and her team, please come to the stage. The second name is Pooja Pal and her team. Please come to the stage. Now I would like to invite Dr. Suchismita Roy, Assistant Professor, Department of Paramedical and Allied Health Sciences, Midnapur City College, and convener of this seminar to kindly come to the stage to deliver the vote of thanks. A very warm welcome to you, madam. Let's give her a big round of applause.
Good morning, everyone. Before I start the vote of thanks, I would like to ask any participating students from Midnapur City College and other colleges to share their experiences about this program. So, come any one student from Midnapur City College. First. Anyone from our college? কেউ আয় একটা দীপ ওই যে আসছে দীপ আসছে and any other one student from any other college, Raja Enil Khan Women's College or Bella College, any students?
First of all, thank you, Midnapur City College, and all the teachers and respected teachers and delegates here. I am came here for 3 times, and I am experiencing very knowledgeable session. I am attending very knowledgeable session, and I am gather many knowledge from the teachers, respected teachers, and I am really enjoying this seminar, and I want to come here every year, and really I am enjoying the all sessions, and my friend come, my research mates, Anonna, Ria, and Pipika, all are appreciating me. My respected teachers, Suchmita ma'am and Shaboni ma'am, and they all are very pampering me as a student. And thank you, sir, principal sir, and Shantanu sir, Kuntal sir. They are helping me in my research also. So I am really grateful to Midnapur City College. And last but not the least, my supervisor, Dr. Dilip Kumar Nondi. I am very grateful to him. And thank you giving me this opportunity in every time to present my work in here, and also appreciating me with the prizes. So thank you.
Good afternoon to everyone present here. This is Deep Chottoraj from the Department of Nutrition and Dietetics, PG, final semester. Basically, I would like to express my special thanks to this place, Midnapur, as well as to my college, as not just joining here as a student and concluding my journey with just the MSc, but also I think the college was a lucky charm to me for some other opportunities that I got. Special thanks to the college, and especially I've never seen such a supportive HOD like I got, my HOD, Dr. Shaboni Pradhan. A poster everywhere. I would like to request to everyone to please provide a big round of applause for her this dedication and that type of management what she can handle. Please do. Even our ma'am, SR ma'am, she was strict but she was also friendly at the same time. I would like to thank her at the same time for guiding me through the lab and all type of possibilities that I could explore. And after going through some of more semesters and all coming across to the research papers and all, I met my true motivation, that is Dr. Shantanu Kaur Mahapatro. I would like to thank him as well. Please do provide a big round of applause for our sir as well for such motivating and such capacity walks. And last, though food wasn't my perspective because I'm into some restrictions from my ethical backgrounds, but the total session was awesome. From the past year to this year, I could see many opportunities to question the judges and to gain some fruitful knowledges as well. The session rolled awesome, and also the students who have come here this time, I would like to express my thanks to all of you for maintaining the decorum of the session as well as entering through the motions. Especially, I would like to thank the college to provide such brilliant opportunities to the students to know more and beyond whatever they are into the cage of books. Thank you to everyone. Thank you to the college. Maybe this was from my end, this was the last conference that I could attend, but I'm really grateful to the institute.
Thank you, Deep. Now, this is the time for the vote of thanks to the participants and everyone. So, good morning, everyone. I hope you all have a nice experience at Midnapur City College. And on behalf of the Midnapur City College and the organizing committee, I extend my most sincere thanks and gratitude to all the participants, guests for their presence in these 3 days programs. And it's my privilege to propose a vote of thanks speech and acknowledge all the contributors of this program who has worked really hard to make this program successful. And we are at the end of the 3 days session, and the conference theme was the Biotechnology for the Healthier Life at the Midnapur City College MCCCON 2025 between 4th to 6th February. And I am very much thankful to our sponsoring agencies, DST ACRB, Department of Biotechnology, and CSIR, Government of India, for providing fund to conduct such conference, and it would have been impossible to organize it without their support. And we are immensely thankful to our Honorable Director Sir of Midnapur City College, Dr. Pradeep Ghosh, for being the energetic person to stimulated us to do our best and standing as pillars of strength. Without his guidance, such program cannot be successfully conducted. And I would like to thank our principal, Dr. Sudipto Chakraborty, Midnapur City College, and Dr. Kuntal Ghosh, Vice Principal, for their support and guidance to conduct such a nice event. And I would like to thank the coordinator of this program, Dr. Dilip Kumar Nondi sir, for his constant support and guidance to conduct this program. And with a deep sense of appreciation, we thank you, sir, for your untiring effort. Actually, we have learned all these things from you during our PhD and till now. And I would like to thank Dr. Professor Nirmal Kumar Sarkar, Emeritus Professor, Department of Biological Sciences, Midnapur City College, for being present with us in these 3 days and chairing all the sessions. And not thank you to the Shravani Pradhan, my friend, but she is actually carrying everything. She is everywhere to conduct such a nice program, for being every part of the program. So our sincere thanks to the Honorable Vice Chancellor, Professor Sushanta Kumar Chakraborty, Vidyasagar University, for his presence and valuable words. For the participants, I would like to thank Dr. Abhijit Rai Chaudhuri, Inspector of Colleges, Vidyasagar University, and Professor Devidas Ghosh, former Dean and Professor, Faculty Council of Postgraduate Studies in Science, Vidyasagar University, for their active presence. And then I would like to thank our distinguished scientists and speakers and guests, Dr. Sanjay Bosak, Scientist F and Deputy Director, Molecular Biology Division, ICMR, National Institute of Nutrition, Hyderabad, Dr. J. Srinivasa Rao, Scientist F and Head Food Chemistry Division, ICMR, NIN, Dr. Arnab Sen, Professor, Department of Biotechnology, Bioinformatics Facility Center, Vishwabangla Genome Center, University of North Bengal, Dr. Sanjay Ghosh, Professor, Biochemical Engineering, IIT Roorkee, Professor Dev Prasad Chattopadhyay, former Founder, Director and Scientist G, ICMR, National Institute of Traditional Medicine, Dr. Shobhik Mukherjee, Associate Professor, BRIC, National Institute of Biomedical Genomics, Kollani, Professor Anant Kumar Ghosh, former Professor, Department of Biotechnology, IIT Kharagpur, Professor Keshav Chandra Mondal, Professor, Department of Microbiology, Vidyasagar University, and Dr. Kaushik Das, Assistant Professor of Nutrition, Bella College. And we would like to thank the young scientists who presented here, Mrs. Soumi Pal Chaudhuri, Assistant Professor, Department of Nutrition, Mohishatul Girls College, Dr. Mohammad Rehan Uddin, Assistant Professor of Physiology, Raja NL Khan Women's College, Dr. Kuntal Ghosh, Assistant Professor of Microbiology and Vice Principal of Midnapur City College, Mr. Sudipto Mongol, Assistant Professor, Department of Pharmaceutical Sciences, Jharkhand Rai University, for their nice presentation in this conference. Our special thanks to all the participants, teachers, students and research scholars coming from different colleges and universities for their participation and presenting poster or oral presentations. We would like to thank Dr. Orpita Raj, Abhishek Chakraborty, Bishodev Rajbanshi for their nice representation and active engagement to conduct such program. We thank the research scholars of paramedical and allied health sciences, biological sciences for their constant engagement for the success of this conference. We thank to all the staff of administration, Mr. Abhishek Das, Momita Roy, Shumon Mullik, Haru Patro, Shubhomoy Das, Arindam Shahu, Shubho Mal, Shumon De, Jagannath Samanto in charge of exam cell and all other staff for their support to conduct such program. We thank to all the students of nutrition who volunteering in these 3 days and participated in this cultural event. I would like to thank all the members of the organizing committee for their contribution to work this really hard for them to make this program successful, and also we thank to all the faculties and staff of Midnapur City College. I would like to thank the food providers, decorators, sounds, camera, transport and media persons for helping in the smooth conduction of this program. And I would like to thank all other sponsoring agencies for their support. And last, on behalf of the Midnapur City College and organizing committee, I would like to say that all the participants that in this program, if any mistake or anything wrong is done with us or to the participants, please apologize us for that and accept sorry for that. Thank you. And with this vote of thanks, I think the program is at the end of this conference, and then we will start the Foundation Day program at the open stage. Thank you. Student come to the stage for a group photo.
Quick, quick. अरे आए ना रिसेस করে চলে আয় তোরা চলে আয় আয় তোরা আয় আয় দাঁড়িয়ে